Hi everyone, and welcome to our second day of Varsity Sci. And what we will have this afternoon already actually are two great talks on ecology. So our first one will be from Margaux on uncovering the genetic diversity of coral reef communities in the Indian Ocean. And that will be followed by Alec, who will be talking about using scientific evidence to save species and the problems and solutions associated with that. So without any further ado, I will pass on to our first speaker, Margaux.
Can everybody hear me? Great, okay. Hi everyone, my name is Margaux. I'm a second year PhD student at the University of Oxford and the Institute of Zoology at the London Zoo. I'm going to talk to you about my PhD project today, which is focused on investigating the drivers and the diversity of coral reef cryptofauna communities in the Indian Ocean. So a little bit about me first. So I did my bachelor's and my master's at Imperial College London. I'm particularly interested in biodiversity, both in marine and terrestrial ecosystems, its patterns, its functions, and the consequences of its loss. So before I start talking about my PhD, I just thought I'd talk a little bit about biodiversity loss, first of all. So biodiversity loss, which is the loss of species, is currently happening at an incredibly frightening rate due to our impact on ecosystems around the world. Biodiversity loss is now recognized as, by international agencies and governmental bodies, as a priority. But much more work needs to be done in order to successfully mitigate the loss of species. For example, the UN is about to do a summit on biodiversity at the end of this month. And recently, if you haven't watched it, and I really recommend that you do, David Attenborough released a new documentary talking about biodiversity and biodiversity loss. And I think it's a really great way of introducing the concept of how dependent we are on ecosystems and how ecosystems function with biodiverse species populations, especially in a day today, like in the midst of a pandemic, and how that is related to the loss of biodiversity. So I think personally, there are three things that we need to tackle in order to mitigate biodiversity loss. First, it's a better understanding of the patterns and the current numbers of species that we have across various ecosystems in the world. Two, how we can run effective monitoring programs. We need to keep an eye on species levels and patterns as we aim to conserve, to manage, and also to avoid altering ecosystems even further. And finally, how to mitigate and protect the species that we already have before we lose any more. And I think that comes into implementing laws and policies based on the data that we collect in order to restore and protect habitats in a sustainable way. So I'm particularly interested in the biodiversity of marine ecosystems. Whilst my bachelor's and my master's were not in marine sciences, I tried to always steer my coursework and my projects towards more marine ocean science. And before I talk to you a bit about my PhD project, I thought I would take the time to just plug in my latest paper that I just released in the Journal of Applied Ecology. Essentially, it's based on work that I did for my master's project. And essentially, it compares a DNA-based method with a morphology traditional survey method for identifying invertebrates in a low diversity, polluted estuarine ecosystem in the UK. So it really was from this project that I became particularly interested in the hidden, the small scale diversity within marine ecosystems. And it really highlighted for me the first time how much of a gap there still is in some parts of our knowledge of taxonomic groups, their populations, and how to monitor them. So that leads me on to my PhD. Like I said, I've just completed my second year. I have 4 supervisors, 2 of which, Mike and Catherine, are at Oxford, whilst Emma is at Imperial College and Chris is at the Zoology Society of London in Regent's Park at the London Zoo. So my PhD project is funded fully by the Bertrelli Foundation in Marine Sciences. Overall, I'm part of a team that is trying to ascertain 3 things. First, we're trying to identify how diversity is distributed across reef systems in the Indian Ocean and their connectivity. So we're trying to study networks of genetic connectivity across different ecosystems there. And finally, we are trying to understand what environmental variables might be driving local diversity patterns. So as part of this group, I study the diversity and community patterns of cryptic invertebrates. We also call these cryptofauna. So what are coral reef cryptofauna? Well, they are the small, hidden and encrusting organisms that live in and amongst the coral matrix. This includes sessile organisms like sponges, bryozoans, tunicates and cnidarians, as well as mobile organisms like crustaceans, worms and mollusks. Although they're mostly inconspicuous, they actually represent the majority of metazoan coral reef diversity and are woefully understudied in comparison to more conspicuous groups like corals and fishes. They play really vital roles in coral reef ecosystems by acting not only as nutrient cyclers, as filter feeders, as detritus eaters, they also are a large part of the feed of prey for invertebrate reef fish. Whilst they're underrepresented in traditional survey studies, new genetic tools and sampling methods are slowly allowing us to get a bit more of an understanding and to sort of access that ecosystem that otherwise is really hard to work in. So where do I work? I work in the British Indian Ocean Territory, which, as you can see, is in the middle of the Indian Ocean. It's very remote. A marine protected area was declared there in 2010. And fishing is illegal there, pollution is very low. Essentially, it provides quite a unique opportunity to study reefs in a somewhat near pristine condition with human impacts removed. A large proportion of the scientific work in the British Indian Ocean Territory focuses on corals, fish diversity, seabirds, and the movement of large predators and turtles. But actually, nobody else at the moment is looking at benthic invertebrates or microbial diversity. Benthic invertebrates, unfortunately, like I said, are quite small, they're quite difficult to sample, and since they're affiliated to both live and dead corals, it can be really difficult and destructive to sample them, and especially to standardize their collection. So, in order for me to do my project and minimize the impacts of my work, and also to standardize my sampling, I use artificial structures, aka called arms. These are simple. They're made of 9 stacked PVC plates with gaps in between them that essentially serve as a substrate with lots of varying microhabitats for sessile and motile organisms to colonize. So they're gradually being used worldwide, and they have already been shown to be an ideal sampling and biomonitoring tool for organisms to colonize. But currently, nobody else is really using them to study Indian Ocean coral reef diversity. Like I said, they're highly standardized. They all look the same, meaning that the samples collected using them can be compared not only across geographical location, but also across timescales. So how to use arms to sample reefs? It's really simple. First, you put them together, and then you attach the devices directly onto the reef matrix using rebars and a lot of zip ties. You then try and leave them fixed and undisturbed for a period of time, which can range between 1 to 3 years. And then during that time, you'll have sessile species which will colonize and grow on the plates. And in turn, that will start to create habitat for motile species to come and colonize as well. So different studies have analyzed arms data differently, but most of them use both DNA and photoanalysis to identify the collected specimens. So overall, for my PhD, firstly, I'm trying to assess and catalog the baseline diversity of cryptic invertebrates in the British Indian Ocean Territory. I want to know what the diversity is there, how much of it do we already know using genetic tools or photo analysis tools, and how much of it do we not know or maybe is currently undescribed. I'm also quite interested in the succession of communities, so I want to know over time which taxonomic groups are perhaps key colonizers and how that community might change over a 3 to 4 year period. I'm also interested in investigating what environmental variables might be driving local diversity patterns across different reef systems. So for example, between lagoonal reefs, seaward reefs, exposed versus sheltered reefs. I'm quite interested in that. And to do that, I'm working with a team from Stanford who put out really sort of fine scale measuring sensors that collect information on a bunch of variables, including temperature, light, pH, oxygen, currents and tidal wave action as well, at the same sites that I'm trying to collect specimens. And finally, this is a more recent interest of mine. I'm really interested in looking at the microbiomes that is associated with different sessile communities. So by the microbiome, I mean the composition of microbes, which includes bacteria, fungus, viruses, even algae within the sessile communities. Essentially, the microbiome is the powerhouse of coral reefs in terms of the cycling of nutrients. And if we can understand which microbiomes are associated with which sessile taxonomic groups, then maybe we can try and predict the functional output of reefs under different climate change scenarios where different taxonomic groups might become either winners or losers or may genuinely just change. So how am I doing that? So 30 arms were deployed in 3 locations across the northern atoll of the archipelago in 2018. In 2019, I went there and I collected 9 of these, so 3 from each of the 3 sites. I was actually out there this year during the February-March period. I was supposed to be out there for 2 months, but because of the pandemic, we had to repatriate home. And unfortunately, I was 2 days away from my sampling site, so I have no data this year. But the plan is to try and go again next year in a longer expedition, and then hopefully the following year after that as well, in order to start building a time series. So I'll show you that in a moment. So just before I go on to like some of the results, I wanted to show you a little bit where I work. So the top photo on the left here is the patrol vessel that we do our expeditions on. And then the bottom left is an example of a smaller craft that we usually take out on a day's dive. So a typical day on board includes an early morning dive, so for me to retrieve one of these devices and potentially carry out some other work like coral collections or benthic surveys, fish surveys, helping out lots of other teams, as there's much diverse work going on in those very sort of short field work periods. And the rest of my day is really spent processing all my specimens back on the back of the boat. So I thought I'd show you a quick video just so that you have an idea of what that looks like and the types of samples I'm collecting. I recommend putting the volume up though, because the volume setting on the GoPro video is very low.
What do we have here, guys? And where did they come from?
So this is all from Elongles. This is this morning's arms data that I just got. And we're just sorting through the volatile organisms. So anything that's 2 millimeters or bigger.
Okay.
Put into individual groups.
Cool, pretty.
Some little starfish, loads of little hermit crabs, some little stars. And next, sorting through, trying to catch all of them. We'll take pictures, really? ID them, and then put them in the freezer.
Ah, and what do the arms trays look like? Do you have any that I can see?
So they're all in here at the minute. We've disassembled the plate. So usually this is the base plate.
Uh-huh.
And then on top of this we have 9 PVC plates that stay out for a year.
Cool.
And currently they're chilling in some cold water.
Nice. And then after this, what happens to them?
So we've separated everything that is water from everything that's sessile.
Uh-huh.
Everything like sponges, corals, algae. We'll be subsampling that later.
Okay. And you're just taking total counts, or are you doing something with DNA?
Yeah, no, so we're going to be sequencing everything individually and then also bulk.
Nice.
Trying to figure out the diversity, how many new species there are.
Awesome.
Anything we don't know yet.
Fantastic. That one's really pretty. Ooh, little shorties.
Okay, so I'm just going to turn my volume back down a little bit. So hopefully that gives you a little bit of insight as to what the conditions are like. I mean, the reason why we go in March and April is because we have a weather window. So most of the time the weather's good. Sometimes the weather isn't, and that kind of changes our plans day to day. So a little bit more about the types of samples that I actually collect from the ARMS devices. It's more than just individuals in little sampling cups. What we get is actually a whole range of stuff. So first of all, I collect photos. I take photos of the plates, as well as large motile individuals above the size of 2 millimeters individually. And I also take individual and bulk samples of tissue from sessile fauna, as well as individual and bulk samples of mobile species, depending on their size, which we split using sieves. I also take benthic transect videos of the surrounding coral cover in order to have an idea of what sort of the live percentage of corals is around and whether that might be influencing what we're seeing on the actual ARMS device. So here's an example of an ARMS device underwater in the forefront, as you can see, and sort of starting to camouflage into the local reef system. And behind we have one of the members of the Stanford team with their array of measuring sensors. So as you can see, the idea of our project here is that really at the same place underwater, we are trying to get an idea of the biological diversity and the environmental parameters at a really fine local scale. So in terms of what we get, here's an example of one of the plates. As you can see, a whole bunch of things is already recruited onto there, even after a single year underwater. We have, sponges and tunicates, as well as lots of macroalgae and turf algae. We've also had quite a lot of polychaete worms, bryozoans, and quite a lot of hard corals and soft corals too. But aside from the sessile groups, we also collected about 500 motile invertebrates, including crabs and shrimps, some flatworms, lots of brittle stars, some sea spiders or sea cucumbers, urchins, and quite a few cryptic fish as well. So before I go through some of my results, I just wanted to explain a little bit more about the type of genetic tools that I use to study my samples. So for the larger individual organisms, I carry out what we call standard barcoding, which involves extracting DNA from a large individual and then amplifying a certain gene region of interest using PCR, also called a barcode region. This small piece of DNA enables us to identify this organism's, hopefully down to species level where the information is available. And then we essentially use a traditional sequencing platform to sequence the PCR products. I also use metabarcoding, which is similar to barcoding, but instead we simultaneously extract and amplify a barcode gene region from all specimens within a bulk sample. For example, like the motile bulk samples or the sessile bulk samples from the plates. We then amplify and sequence the bulk DNA using high throughput sequencing. In my project, I'm targeting a whole range of gene regions, including the COY and 18S gene for eukaryotic diversity, as well as 16S and ITS for bacterial and fungal diversity as well. So following the sequencing, we use a bioinformatics pipeline to try and filter and merge DNA reads, as well as assign taxonomy and create a curated species table. With that, we can then merge both methods' data sets and then have an idea of the alpha diversity within arms or within sites and the community composition between sites and between arms. So I completed the COI and 18S metabarcoding of 2019's samples that I collected. And I've targeted, like I said, both the COI and 18S, but here's some preliminary results of the COI gene sequencing output. So using a strict bioinformatic pipeline, I was able to pull out several thousand amplicon sequence variants, aka ASVs from the sequencing reads. Overall, I detected the presence of 90 genera and 48 species, which were identified across several major phyla groups. Worryingly, we even detected the presence of the crown of thorns starfish, which is on the bottom left of the pictures of species. This could be just a one-off population. We're hoping it is, as this can be a really destructive organism for live coral cover. So you're probably thinking 48 species, that feels really low for a study that, looks at hundreds and hundreds of taxonomic groups. Well, it's true. And the reason why is because one of the problems that we face when we look at benthic invertebrates is that actually the public databases that we're trying to compare our personal sequencing reads against sometimes lack information. And I think that's something that's happened in this preliminary study here. So for example, in gray, you can see that a lot of the sequencing reads have not been assigned to any phyla groups. Also, one of the reasons why I don't want to make too many assumptions on the patterns that we can see in this bar plots is that, like I said, I've only looked at the COI gene, and the COI gene can be biased towards certain taxonomic groups. And so my plan next is to merge the 18S results and rerun a new bioinformatics pipeline to try and have a more accurate view of the actual taxonomic composition. In terms of the community composition, we can see that actually beta diversity across site and across individual arms cluster really strongly. So here the sites are in colors, and each point represents a single arms device across the three fractions that I sampled. So the small motile fraction, the large motile fraction, and the sessile fraction. As we can see, the left-hand plot, which is the small, sort of planktonic size motile fraction, clusters really strongly by site, and so does the sessile groups on the right-hand side plot. Essentially, it looks like community composition is really similar within sites, but differs strongly between sites. So what next? So apart from analyzing that 18S dataset, the plan is to analyze the microbial diversity within those metabarcoding samples from last year using the 16S and ITS regions. I also need to finish the barcoding of the larger motile specimens. This is something I'm doing now that the labs have started to reopen. I have both tissue samples and large motile samples, and so that's taking me quite a while. I'm collaborating currently with someone from the Natural History Museum who has expertise in sponge identification, so that we can try and morphologically identify sponges at the same time as providing new genetic barcodes, in the hope that we can try and fill out some of the gaps that are currently in online databases. For example, uploading our work to the Sponge Barcoding Project. And finally, I really need to carry out the photo analysis from those photos that I collected last year. The idea is to use a random point count system. So you put about 225 points across each plate photo. And with that, you identify whatever falls under, well, whatever the point falls on. And then you get an idea of not only recruitment success, but also taxonomic composition. Well, what will be really interesting is to compare those results with the metabarcoding genetic analysis and to see whether we get sort of similar patterns of community composition. Overall, the idea is to merge all of these three avenues in order to provide the first baseline diversity study of the Chagos archipelago using ARMS. Ideally, another part of this would be to then compare to other ARMS projects from around the world. There's been a lot of work using these artificial devices in the Red Sea, as well as in French Polynesia, and being able to compare coral systems across oceans using a really standardized protocol might give us a really brand new insight into how these different reefs function. Ideally as well, the plan is to get back out there. I mean, currently travel is still restricted. We were supposed to go back on an expedition in November, but this has been cancelled. So the idea is to also spend some time really planning ahead for potentially what might be the last field expedition next year. So quickly, before I finish my talk, I just want to talk a little bit about other things that I'm doing in my PhD project. One of them is a literature review on benthic invertebrates and microbial communities, what we already know about them in coral reefs, their functional roles, and what those communities might look like in future reefs, and what we still need to do in terms of integrating these groups into biomonitoring programs and why we should. And finally, I'm really interested in methods-based comparisons. Like in my master's project, I think that the ARMS are really interesting, but I am quite keen in seeing how they might be biasing diversity that is collected using them. That's why comparing what you get on the ARMS with the surrounding coral cover will be a nice indication of how effective are these in representing the actual coral reef diversity. And finally, one of the bigger aims of this PhD project is to be able to produce more data than what we currently have. So it's a bit of an exploratory study in a way, in the fact that I'm trying to get as many genetic barcodes and hopefully making them open access into sort of a biobank repository specifically for the Indian Ocean, so that people can start comparing and maybe doing some genetic analysis between populations. And that's it. I just want to thank my supervisors as well as collaborators, and as always, everyone who's ever helped me in the field. Thank you. Does anybody have any questions, or Christoph?
Okay, thank you for the talk, and I'm going to run the Q&A session. So if anyone has any questions, they can put them on Zoom or through the YouTube chat. And then, so we have one, what were the barcode regions that you used for the individual barcoding from Zoom?
So for example, for the COI, for the metabarcoding, we used a sort of a shorter version, not the entire full Fulmer region. So using a 315 base pair region, using the primer set MLCOI int F and the JGHCO198, I think it was. That's for the COI, and the 18S, I need to double check, but it was also a shorter region, sorry.
Thank you. And then also from Zoom, how does the proportion of uncharacterized species compare to other comparable experiments?
So at the moment, in order to determine how many of my samples are undescribed, I need to also look at the 18S and not just the COI. So what we get sometimes is you get primer bias. So although the COI region is universal and you do find it across all eukaryotic groups, the primers that you use sometimes were designed using only certain individuals from taxonomic groups that don't represent all of the eukaryotic domain. So you have to use two different types of primers at least. So I think especially the taxonomic groups that I'm looking at, sort of marine versus freshwater, we have a lot less information from Indian Ocean species than we do from Pacific Ocean species or freshwater species or Mediterranean species. So that's one of the problems, is really I'm trying to identify things with very little, previous knowledge. So wherever I can add on to that knowledge would be a useful output. Hope that makes sense.
Yes, thanks. And then from Daniel Lim on YouTube, how did you choose the regions where you set up your arms?
So the, as in the locations?
Yes.
Yeah. So, okay. So the Bertarelli program in marine sciences focuses on the British Indian Ocean Territory. It's a big sort of collaboration of institutions and researchers, and the idea is to survey and study that area as much as possible in order to gain an understanding of what near pristine reefs in the Indian Ocean supposedly should look like. So that's why we chose that atoll, that archipelago. In terms of the locations, we wanted to be reachable. So for logistics reasons, I mean, sometimes if you have 12 teams on a single boat and only 3 weeks to do expedition work in, then if you can all go to the same sites, it makes life a lot easier. But I also wanted to have different types of ecosystems. So we have one lagoonal site, which is really protected, where quite a lot of high sedimentation. We have like a very exposed site with really strong currents and tides. And then we have sort of a semi-exposed side, which is not on the inside of an atoll, but on the outside, but not with prevailing waves and currents. So we're trying to get a little bit of a broad range of local ecosystems. They're all at the same depth, so.
Thank you. The next question is from one of the people who are on Zoom.
Yeah, hi. I just wanted to ask, because your time series data sounds really cool. Have you heard of the BioTime Database?
No, I haven't actually.
So I think they'd be really interested in your data, because they generally look at databases from around the world that are time series, and they look at how biodiversity has changed over time. So yeah, I just thought they might be quite interested in your work.
Yeah, I mean, that's one of the reasons why we wanted to use the ARMS for a time series, is that because it's so highly standardized and you can really easily reanalyze anything, because it's, photos are there, sequences can be reanalyzed using new bioinformatics pipelines. And because they're highly standardized, you can really compare sort of different time points on a time scale. And to my knowledge, no one else has used these devices yet to look at succession or to do a time series. It's usually put them out there, pick them up 2 years later, and that's the extent of the project. So yeah, hopefully it can be contributed in another way. That'd be great. Thanks for letting me know.
Yeah.
Thank you for your question. We have another question from Zoom from Christoph. Have there been tools similar to ARMS used previously?
You mean highly standardized tools for marine biomonitoring? I think that's the reason why ARMS were created. So people have always wanted to standardize the collection of marine specimens, and people have used things like bricks or cements, or like cement blocks have been used as well to somehow create cryptic ecosystems. People have used things like ceramic recruitment tiles to look at like coral recruitments. But the ARMS, to my knowledge, is the first sort of global initiative to make something that is not only simple enough that the protocols are easy to follow, but also somewhat complex enough so that you can try and represent as many different types of microhabitats. So within the ARMS device, we have several microhabitats. We have the top plate, which is completely exposed. Then we have a series of plates which either have an open gap between them or are closed off with nylon spaces. So you get these sort of like 4 little niches. So we're trying, to use them as much as possible and really show that they're a great tool, actually. And because they're made of PVC, the idea is that economically they're quite simple to put together, and you can make them yourself from wherever you are in the world, as PVC is really globally available.
Thank you. There are just 2 more questions. So one is from me. I'm personally very interested in microbiomes, and I was just wondering how you're quantifying that. Are you swabbing the larger invertebrates, or are you just doing a more environmental microbiome sample?
So at the moment, from the 2019 data set, it's very much environmental. It will be from the DNA that we collected as we scraped off the sessile organisms from the plates. But in the future, we were thinking of providing essentially a grid where you swab individual, taxa, like swabbing a certain sponge or swabbing a certain bryozoan, to start looking at like individual taxa-specific microbiomes. Yeah, so that's a plan for the future, but didn't have time last year.
Thank you. And then the last question is from Ella on Zoom, and she's asking, what was the biggest challenge you faced during your PhD project so far?
I mean, so far the biggest is lockdown. I mean, I was stuck out there for an extra 2 weeks when everybody else was already in lockdown in the UK. It was a bit of a mission to come back from a really remote part of the world. But that was quite smooth because we had the help of many other people. Personally, I think the most complicated thing is the time that it takes to organize logistics for expeditions. I spend a large portion of my time in the run-up to the expeditions, spending the majority of my time on posting things or doing medical checkups or preparing, getting things like checked in terms of my gear, and organizing takes a long time. And so there's quite a lot of months that I don't have for writing, which I wish I had. Logistics is what I would say.
Thank you very much for all your answers and the talk. And I'm going to hand over to Christoph now for an introduction of the next speaker.
So thank you so much again, Margaux, for that brilliant talk. I mean, I'm a biochemist, so it's not really my area of expertise, but I really enjoyed finding out more about your work, and also really appreciated the amount of detail, the images and the photos that showed more about your work and the importance of it, as well as the various experimental methods you use in your field. So thank you so much for that again. And we're going to have a talk from Alec next, who is going to talk about the importance of scientific evidence when it comes to saving species and the problems and solutions that are associated with that. So I'll pass over to Alec, our next speaker.
Okay, thank you very much. I'll just set this up. Okay, can everyone hear me okay?
Yep.
Oh, yeah, okay, great, okay. So yeah, I'll just introduce myself a bit. As you said, my name's Alec Christie. I'm a PhD student in the Department of Zoology here in Cambridge. I work in a building which is brilliantly named the David Attenborough Building, and it's home to lots of different conservation organisations within something called the Cambridge Conservation Initiative. And so it's a really great place for conservation. And my background was actually in marine biology. So I studied at the University of St Andrews for a bachelor's in marine biology. I did a lot of work on coral reefs, actually, so it was really cool to hear that talk. And I also did work on marine mammals as well. So a bit of a change now. I've sort of gone to a much sort of more global scale, just looking at evidence-based conservation. so I'm just going to talk a bit about my work, my PhD work, and some of the problems and solutions we face in making conservation a more evidence-based discipline. So I know you probably don't want to hear about COVID-19 anymore, but I'm going to say about it anyway. I think it's been really cool that, you know, on a day-to-day basis now, we are talking about scientific evidence and its use in practice and policy. And there's a long history of actually evidence-based approaches in medicine. It was started in about the 1960s or 70s by someone called Archie Cochrane, and he sort of led the way in getting doctors to look at scientific evidence and to actually test whether different treatments work and how effective they are and what side effects they have. So evidence-based medicine is quite a fully fledged field now, and so if you go to your doctor, your GP, you will get treatments recommended to you and given to you that are backed up by the best scientific evidence. And so what I'm interested in is, why can't we do this in biodiversity conservation? Why can't we prescribe treatments to conserve species using scientific evidence? And so there's a few reasons why we aren't doing this already, because you may think, you know, why haven't we been a bit more proactive at this? Well, conservation, you'll know that it's probably very chronically underfunded. There's also been quite a big focus on threats rather than solutions. So there's a lot of research that goes into quantifying how much habitat degradation there is, how many species we're losing, but not actually how to actually combat that, how to tackle those threats. There's also a perception that sort of on the ground, that ecosystems and species interactions are so complex that and the habitats are so different that we can't actually generalize. So we can't take the scientific findings from one study in a different part of the world and apply them very well to another part of the world or another species. So that kind of perception is a problem, and that hasn't really been properly tested. There's also an issue when conservationists and organisations are pretty bad at publishing when they fail. So when actions don't work, there's often very little motivation to actually publish these findings. And that's quite a general issue across science, this issue of publication bias, that people publish, there are positive findings that are overrepresented in the literature compared to negative or neutral findings. So that's also a big issue. And finally, I think there's just been a fundamental lack of collaboration between scientists and actually conservationists in the field. So actually bridging that gap between science, academics, and practice and policy is a big issue, and it's a big issue for lots of disciplines. But evidence-based medicine has done quite well at connecting those two. So there are approaches that in conservation, and I'm going to talk about this one through which I'm affiliated, which has been going on for the last few decades, and it's called conservation evidence. And what this project does is it takes the scientific evidence and summarizes it on this website, so that for every conservation action possible conservation action you can think of, the researchers go out into the literature and find the studies, and they summarize it on these different pages. So you can search for different conservation actions like this one. There's different summaries of the studies, which you just saw at the bottom there. And then for this action, there's then a key message section which tells you, okay, how effective is this action based on the scientific evidence, based on what we know. So this is a really great resource. It's completely free online at conservationevidence.com. And they also get experts to assess that evidence. So you can see on the right, it's assessed for its effectiveness, the certainty of the evidence, and also possible harms of this action. You know, could it actually have negative impacts? So the database is growing all the time. It covers lots of different areas, birds, amphibians, mammals, also benthic invertebrates now, and it's got kind of growing into the marine realm, but lots of different areas. And so I'm going to talk a bit about my work, which has been to look at the underlying data in that database, these studies, and look at the gaps and biases in that literature. So where are we testing conservation actions, and where are the gaps and biases that we need to fill? So I'm just going to first show you this, just going to move this out of the way. I'm just going to show you this map, these maps first of all. So this is a series of maps that look at the distribution of studies across the world. So this is for amphibians. So if we just look at the top left-hand map that says all designs, that's all the studies in the world that have tested conservation action for amphibians. And you can see there's already big gaps. There's gaps in Africa, there's gaps in vast parts of Russia and Asia, India, South America, and there's big concentrations in the US, Europe, and to some extent Australasia. And then if we look at the rest of the maps, so what these maps are showing are for studies that have used different experimental designs. So the bottom two experimental designs, I'm not going to go into detail on what they are, but you can consider those as more robust, more rigorous than these other designs. So those bottom two maps, you can see actually the studies that are quite rigorous, that are rigorously designed, there's actually even bigger gaps. There's big holes in the evidence base, and they're very geographically restricted to North America and Europe. Whereas for some of the simpler designs, the less rigorous ones in the middle and the top right, there's generally slightly more of them, and they're slightly more distributed around the world. And we see the same for birds. So there's more studies on birds, but generally there are similar gaps and patterns and biases here. So clearly, where we're conducting studies already, we can see there's a bit of a problem. And an interesting way of looking at this is to say, okay, in those locations where we're testing studies, how many species are there in those locations that are threatened or data deficient? So threatened species are classified by the IUCN Red List as either vulnerable to extinction, endangered, or critically endangered. So what we've got here is in these different locations, we can see how many studies there are in these sort of grid 2x2 grid cells, and then how many threatened species or data deficient species are there. And generally, so for amphibians, there's a negative relationship. So we're actually conducting more studies in places with fewer threatened species, which is pretty odd and completely against what we're supposed to be doing in conservation. And for birds, there's a similar pattern. It's quite weak, but there is a statistically significant relationship there. And for primates, there isn't a relationship. But either way, whether there's a relationship or not, what we want to see is actually a positive relationship, that we're testing more, we're doing more studies testing conservation actions in places where there's more threatened species, and that's not happening. So that's a problem. So moving on to away from the geographical biases to taxonomic biases. So this is a phylogenetic tree of birds, of bird orders. So and what we can see is the darker red colours are groups that are underrepresented relative to the number of threatened species that they contain. And if you look at the names in red, those are actually whole taxonomic orders for which we have no studies at all, no studies testing any conservation action. So we can see, hornbills and hoopoes, they actually don't have any studies. Nightjars, hummingbirds and swifts are underrepresented. Songbirds, which is surprising, just simply because of the sheer number of threatened species, they are actually underrepresented. So again, lots of biases, and we can see the same if we look at primate families. So here, the symbols that are in white show families that don't have any studies. So we've got tarsiers in Southeast Asia and night monkeys in Central and South America that actually we don't have any evidence, no scientific studies that test whether conservation actions work to protect them. And we've got a few that are in red or orange that are underrepresented, but then actually we've got quite a few that are overrepresented. So we've got the great apes, the hominidae. They're quite charismatic, so it kind of makes sense that we may be overrepresented the number of studies that are testing these relative to the number of threatened species. So now that's all the biases and gaps that are important. But a key question that we need to ask ourselves for evidence-based conservation to work is, how many relevant studies are there? So practitioners in the field are really keen on evidence that is perceived as locally relevant. So does it test a similar taxonomic group or species, and does it look at it? Is the study conducted in a similar habitat or location? So was that conservation action tested in a relevant context? And they would also be interested in the impact metric that was used. So how did they test? How did they measure the effectiveness of that action? So here, for example, we've got maybe you'll create a pond. So you could measure the success of that in terms of the number of species, or in terms of the diversity of the frog species, or in terms of the reproductive success of those frogs, or just one frog species. So there's lots of ways of measuring effectiveness. And all of those factors that I've talked about will influence how relevant a given study will be to informing a practitioner. So one way I decided to look at this was to say, okay, practitioners could be anywhere in the world. Let's see how many studies are within a given radius of their location. So I've randomly spaced out these, well, regularly spaced out these coordinates throughout the world on terrestrial land masses and islands. And then I looked at, okay, how many studies are there on average for each conservation action? So that's the y-axis. And obviously you'd expect there to be a big decline, but actually what we see is that if you look on the x-axis and you go down to 2,000 kilometers, which is still a massive radius, a massive area for you to look over, there's still only around one study per conservation action on average for a given practitioner. So that means that really there's very, very few studies that are likely to be in a local area to that practitioner. So there's often going to be very, very few studies that they will perceive as being locally relevant. And again, we can ask this question in a more detailed way by looking at different criteria. So a practitioner might be interested in sort of three important aspects: the biome or the habitat, the taxonomic group, and also the metric. So the metric of effectiveness. So those three factors are important. And what we can do is we can look for each conservation action, what was the most commonly studied biome, taxa, and metric, and then how many studies were there that tested those three. So in a sort of stepwise fashion. So what we find is, obviously, as we get more and more strict in our criteria, that the studies have to feature, that they have to contain, we eventually find that this is for the most commonly studied context, that there's still on average less than 2 studies that might be considered relevant. And if we look then of how many of those use sort of robust, rigorous experimental designs, we find that there's obviously very, very few. And this is for the most commonly studied contexts. So if you look at prioritizing threatened species, you'll find that these patterns are far more severe, so that there's almost no studies that have relevant evidence that meets all 3 of these criteria. So that's that message, that there's really very few relevant and robust studies for practitioners. So what do we do? How do we best use a patchy evidence base like this to inform local conservation action? And you can think about this in terms of a bit of a trade-off. So scientists often really want to do big global analyses, big large analyses, and find generality and patterns that explain different things. And they're often concerned very much about the rigor of those studies. On the other side, you've got practitioners and policymakers who really just want local relevant evidence. They're maybe not as concerned about how rigorous it is, but they just really think that it has to be relevant or they won't actually use it. They'll just rely on their own local knowledge or local experience, which it might still be useful, but we can't really know how reliable that is, and also it risks not learning about successes and failures from other places, from other locations that actually is useful. So a compromise is required. So we could just go on with the status quo as it is at the moment, but we focus, we do evidence-based conservation by just looking at the few, if any, locally conducted studies, or we just let practitioners rely on their own experience and beliefs, and we say evidence-based conservation using global evidence isn't possible, which is a bit defeatist. But I think there's the way forward is definitely co-assessment, and what that means is it's weighing and assessing that global evidence base based on its quality and its relevance. So I'm going to just quickly talk through a tool which will hopefully explain that a bit more. So the idea is that what we want to do is take global evidence and apply it to local conditions. So what we would first do is look at the user's local context. So a practitioner may say, I want to know whether this will work, this conservation action will work for me. And they may have a particular taxa that they're going to focus on, and they'll be based in a certain habitat or location. So you can collect that information from them. You can then look at all the studies that have tested that conservation action, and you can extract the measures of effectiveness, their results. You can then first weight these results by study quality, so you can give greater influence to studies that are more robust. And that's commonly done in meta-analyses and things. But then you can also weight studies by their relevance, so you can give more influence to the study results that are from places that are relevant to that practitioner. And that will mean that you end up having different studies have different influences on the final result, and that weighted result should then tell you how likely a conservation action is to work for a given person, for a given practitioner, for a given species in a certain place. And we're trying to work on other decision support tools kind of like this that also integrate local knowledge and experience and also cost effectiveness, which is really important, and bring those together with the scientific evidence so that practitioners can transparently show how they came to a decision. So I'm just going to finish and summarize. So hopefully I've shown you that generally we really do lack relevant and robust evidence to save species from extinction. We focus too much on the threats, and we really need to start looking at how are we going to solve this extinction crisis. The evidence base clearly needs improving. We need to do more studies, but we also need to prioritize this. You know, conservation is chronically underfunded, so we need to make sure we prioritize which species we're testing, where we're testing these actions in the world. And in the meantime, that's going to take a while. But in the meantime, to make decisions now, we need to locally co-assess that global evidence base to inform local conservation action. So I'll just finish there and say thanks for listening, and to my supervisors, the conservation evidence team, and also NERC for my funding. Thank you very much. Any questions?
Thank you for that talk. I'll be taking questions again. So the first question is, in these studies where you're looking at the evidence, do you also consider effects after so-called successful conservation? So for instance, with the black robins on Chatham Island, where it was very successful in bringing back numbers, but then you had the problem where they were the eggs were rolling out of the nest because they'd been pushed back by conservationists. Are things like that being taken into account?
Well, that's a really good question. That is a problem. That is a problem in terms of the studies themselves and the time scale over which they consider, how you define conservation success. And that really has to be, that's why it's so important to assess the evidence for its quality and assess the evidence as a whole and say, well, actually, these studies were quite short in their time scale. They didn't actually consider what happened later on. And it might be that studies come along later on that you can add to that evidence base and say, well, actually, this was shown to work on a short time scale, but actually in the long term, it wasn't successful. So that would also come back into that and sort of address that. But that is a problem. That's a problem with lots of ecological studies, the time scale over which you monitor. So yeah, it's a good question.
Thank you. So as before, if you're on Zoom, you're welcome to ask in person or put a message into the chat. And any questions in the YouTube chat can also be asked if anyone has more questions.
I have a quick question. So you're focusing on kind of deciding which interventions are useful based on existing studies. Do you have any idea on how we would go about deciding which species to focus on? Because it looks like we can't save them all. This is horrible, and this is more of a moral question, I suppose. But do we have any sort of way of gauging which species are more impactful on which species we really need to save, or because they have a very important function in the ecosystem?
Yeah, so no, that's a good question. Yeah, that is a moral question for all of conservation to consider. Yes, there's a lot of work. Well, there's obviously a lot of work on this idea of ecosystem engineers and the relative roles of species. Are some species genuinely more important than others? And given we are chronically underfunded and we have to prioritise, I think it is, from my perspective, I think it's the only way forward is to prioritise. And the IUCN and different organisations will do that and do that in their work. So there is definitely prioritisation going on. And I think there have, off the top of my head, I can't think of any studies, but I know that there must be studies out there that have looked at that sort of idea of prioritising where we put our funds into. And there's also been more global analyses on where you spend the money. So actually, where does your conservation, where does your money bring you the biggest benefits for conservation? And those analyses are really important and powerful, and actually show that in a lot. It wouldn't actually, if we actually allocated our funds to particular countries with particular governments and different levels of different levels of governments and attitudes towards conservation, then actually we wouldn't actually need to increase our funding that much to actually protect quite a significant amount of species. So yeah, there are a lot of inefficiencies at the moment that people are definitely working on. It's a big priority.
Thank you. I have another question. I think nothing else is in the pipeline right now.
Okay.
So this is, I guess maybe Margaux can also chime in on this. I think she's still on the call. I'm more in the engineering department, currently focusing on a new GPS receiver, which is specifically meant to help conservation biologists. So you talked about why we have such a lack of good conservation studies, and you talked about lack of funding and publication bias, and just also the fact that many studies, when they happen, are badly designed. Do you think that, so let's say we have a conservation biologist who's super motivated and they're willing to publish even if their result is negative, and they really want to get to a study. Do you think that lack of affordable, accessible tech is a problem for them often?
That's a good question. Yeah, I imagine that's definitely a barrier. Definitely one of the many barriers to sort of publishing good studies. Yeah, and I can see that often does limit how the quality of the studies as well, in terms of, very often when I'm talking about experimental designs, the more rigorous experimental designs are conducted over longer periods. You need more resources. The logistics are much more difficult. Sometimes it's just not possible, because a lot of the better designs also rely on, you need to know that the conservation action you're sort of testing is going to happen, and often that happens. Often we do a conservation action and then actually let's monitor afterwards, whereas actually planning the action and then testing it as a kind of a whole program is much better. But yeah, funding is definitely a problem, and I can see that that is an issue for conservation in terms of getting that technology and being able to test it in that way.
Thank you.
Okay.
Thank you. So we have 2 more questions on Zoom. One is, has your work been used in practice so far? So is there any study that's worked off that evidence-based conservation so far?
Yeah, so the Conservation Evidence Project actually has these different organisations called evidence champions. So they're organisations that actually have taken it upon themselves to invest in looking at the scientific evidence from the website and using it, integrating it into how they make decisions. So that's really great. There's also, what else was I going to say? There's also, we're publishing a lot on these issues, and journals are now starting to mandate that when you publish a study in a conservation journal, you look at the literature beforehand to see if anyone's tested it. So we're not wasting resources through, just repeating studies is useful, but we're not sort of trying to reinvent the wheel and sort of repeat what we've done in the past when it's not going to be useful. So yeah, there is a growing movement to make this more mainstream, and because of the building we're in, we have a lot of good contacts with different organisations. So slowly but surely, there is a movement towards more testing and more integration of evidence in how they make decisions. So that's something to be optimistic about.
Yeah, that is nice to hear that it's moving towards that then. Another question from Zoom from Margaux is, hi Alec, have you looked at the human aspect of conservation in your modelling? aka how to prioritize conservation actions or areas that include both species conservation and human livelihoods. In the shark illegal trade, we've seen that a great way to conserve species is not only to increase patrolling or fines, but also to work with local populations to find livelihood alternatives.
Yeah, no, that's a great question. And fundamentally, yeah, we're trying to analyze the evidence on more social interventions where you actually, rather than sort of doing sort of traditional conservation where you put a protected area around something and try and protect it, or you do fines or you do poaching patrols, that you actually educate or you engage with local communities, and that's something we're trying to develop. I guess part of the issue with that is that what the Conservation Evidence Project looks at is studies that have quantitatively tested the effect of conservation actions. So it is slightly out of our remit in terms of looking at studies that are more qualitative, and that is an issue, and that does need looking at in terms of how we can integrate that evidence from maybe more social science, more humanities subjects on the effectiveness of conservation actions. But there's it's being increasingly realised that the human aspect is really important in evidence-based conservation. We're sort of working towards that and trying to integrate that more into the scientific evidence and looking at more human-focused interventions.
Thank you for your answers and your talk. And then if there are no more questions, I think I will hand over to Christoph for the summing up of this session.
So thank you very much, Alec and Margaux, again, for your talks. What I really enjoyed about this session personally is just how much discussion there was after the talks and how much engagement there was from both of the speakers and from people on both the Zoom live, both the Zoom chat and also on the YouTube live stream. So thank you for that very engaging and interesting and informative session. And I hope to see everyone soon for our next session of the day. Okay, so I've just finished the live stream. Thank you everyone for coming.